A few left. Just to comment on this, this is the analysis that we're going to do tomorrow, more or less. What we said is how to bring science into the lab. This is one of your assignments. This protocol that you might have gotten from a colleague, in this case me, needs to be modified. This is not the protocol that we're going to use tomorrow. I'm going to explain to you in a moment how this works, okay? So, a part of my research is on the neuromuscular junction. Well, there's neuro in there, so that's why I'm working in this department. And one of the most important things for you to know, neuroscience is not only this bean-shaped organ structure in your head. Neuroscience is also about all the nerves that you have in your entire body. So neuroscience doesn't stop there. It's of course the biggest location of accumulated nerves, but there's lots of nerves in the spinal cord, as you all know, and the nerves extend in all of your, in the rest of your body. And they have important functions there, so that's important to consider. When you have a problem with your nerves in this part of your body, you'll probably end up being at the neurologist as a patient. If you do research, of course, on the other hand, you will be in the neuroscience department or the neurology department. If you have a problem here, it can be either neurological, if it relates to your movements or your senses, like pain, like temperature, and so forth. It can also relate to your behavior and your cognition, and then you might end up in the psychiatry. What is important for you to know is that the fundamental mechanisms that underlie all of these problems can be pretty much the same. And that's important for you to know because you're the ones that study the biological mechanisms of disease. And biological mechanisms of disease can be only a few in general. Either they can be genetic. I'm telling a few things that are not in the slide, so you might want to take a few notes. This is not going to be part of the exam, but it's nevertheless maybe relevant. So disease in general can be genetic, like be congenital, be present from birth, or determined at least at birth. They can be trauma. It's clear if you have an injury that can impair your function. It can be autoimmune, so your own body attacking itself with the immune system. That relates to inflammation. And it, of course, it can be infection, like or a toxic agent. Yeah? So microorganisms that destroy your tissue. So those four are, I believe, the main causes of disease. And they don't only relate to neurology or toxicology, they relate to all diseases in general. So wherever, if you're working on the gut or on any other organ, you have to think of those mechanisms of disease. When you'll be working as researchers, you'll probably focus on one of those. Maybe you do mostly immunology, maybe you do mostly electrophysiology. That is very much specialized. But there are quite a few doctors amongst you, future aspiring doctors. They need to think of all these possible causes of disease. And they translate, because we do translational research, into the basic research. So, if it's neurology, it relates to movement. And then the most important nerves to consider are the motor nerves. Well, these motor nerves are located in the spinal cord, here, and they all the way extend. They can be 2 meters long in some of us, or 150. In other animals, in a giraffe, for example, they're even much longer. But it's a single nerve extending from the soma, which is in the spinal cord, with the axon that can be even 2 meters long, into the peripheral organ, typically a muscle. It goes also the other way around. Then you are related to the senses, pain, pressure, heat. That is also a neurological problem if that is not functioning properly. What I'm going to talk about a little bit today is an example of a disease and take that into basic research like I'm doing. So just for you as an example, how this is typically done. So, in general, what tools do we have available to quantify any disease, any mechanism? We need to measure it, right? The scientists calculate things, they predict it, and after that they do experiments and to measure what's going on. Well, most of what we measure has to translate in something that we see. So our eyes are important. So we can measure everything that we can see, basically. On other organs we don't relate so much. We might measure something by hearing. We might measure something by feeling, temperature, structure, very little. Even less by smelling. Most of our equipment in the lab is based on something that we can visualize. So therefore, the things that we are going to do in this first week of the minor course are measurements that are related on vision. And when you think about it, for example, immunohistochemistry, have you done something like this before? Immunofluorescence in a practical way? I guess yes. Well, it is based on color. The color is translated into a picture, in this case, false color, with a camera. So the equipment that needs to do that we're going to use needs to do another thing. It needs to translate the optical information that we can see into an electrical signal. That's what we do, for example, with the PCR machine. So the first step of the typical assays that we use in biology are optical signals that are then subsequently translated into an electrical signal, so the computer can measure it, or any other electrical device. So, in biomedical research, a lot of imaging, a lot of techniques that are based on light emission, and but of course we want to make the coupling to the actual disease molecule. So how do we do that? One very important aspect of that, one important tool, are proteins and in particular antibodies. And this is, from this I'm going to take an example for the first week of the mind. In some very few cases you can directly measure electrical activity. For example, all of our muscles, for contracting, they produce electricity by exchanging ions over the membranes. So if I do this, very simple movement, there's on the muscle, there's a current that goes along. You can measure it, it's millivolts. So it's a signal that can be picked up directly from the muscle into an electric machine without intervening in any way with something visual. If we want to see it, judge it, we have to, again, modify these currents into something like a graph, like this. So it's important to know that how these analysis systems work, generally based on optical signals transferred to an electrical signal. Very much what something that we do is in electric signal is like a Western blot. Yeah? You get bands, we can measure the size and the position on the gel or on the nitrocellulose paper. We use microscopes that also visualize information. We have OD fluorescent readers and so on. So where does this molecule come in? Well, this is a very important molecule. You all know it. It's an antibody. And why is it so important for all of you? I think all of you already use it. I use it a lot. It's a very important molecule for immunology and also inflammation. Despite the fact that some of you don't really like it, you will have to face some elements of immunology, and at least for the research tools, it's important. So we use antibodies, for example, for immunohistochemistry or for immunofluorescence. We use it for Western blot. In this particular practical, we're going to use it for ELISA. So for that, it's crucially important to understand the properties of antibodies. How can you use it? How can you use these molecules? There's another aspect, and it's more applied research, translational. You can use antibodies for passive immunization. So by directing an antibody with a particular affinity for a virus, for example, you can prevent a disease. For example, if you want to book a last-minute trip to Morocco and you didn't have time to get your vaccine shots, at least against hepatitis, you can partially protect you by getting a passive immunization from the pharmacy, from the travel clinic. So that's an example of a direct injection of antibodies that will protect you for 2 or 3 weeks. You can induce antibodies by vaccination. If you inject a protein into an animal or a human, it will mount an immune response. Part of the immune response will produce antibodies, and that can neutralize the respective disease that you are trying to prevent. It comes, of course, the term from vaccinia, injection, so against the flu. But vaccination is used much more general, and even this passive immunization is using the particular immune response that produces antibody. Now, antibodies are also important for the understanding of a couple of diseases, because they're important in autoimmune diseases. That's one of the 4 characteristic disease mechanisms that I mentioned at the beginning. They are important in allergy. And in general, they are important for inflammation. So, as I said, even if you're interested in neuroscience, you probably don't get around part of the immunology because the immunological tools that are available are important for all disease areas. Yeah?
I bet that most of the students, they don't have anything against immunology. They have something against like metabolism and diabetes in general.
Right. You had too much.
We had that too much before.
Yeah.
Immunology is completely, I think.
Basic immunology is okay with it. Yeah. Okay, that's cool. Thanks for pointing that out. I felt attacked because I'm an immunologist. That's a joke. So, as I said, if you want to use these molecules, which are very useful, you need to understand their properties. You need to understand if they're specific. You need to understand what the affinity is, so how strong they bind, how fast they bind onto it, and how long it takes them to fall off again. How stable they are, because not all antibodies are equally stable. And you need to understand the effector functions of an antibody. So effector functions are, for example, the binding to Fc receptors, the activation of the complement system, and so forth. There's going to be a little bit more about that later. So going back to neuroscience a little bit, it started, the discovery of the most important early discoveries in neuroscience was actually on the peripheral nerves. There were muscle nerve preparation by Luigi Galvani. And he discovered that electrical stimulation of muscles and nerves will lead to muscle contractions. And this is, of course, a visual clue of how something works. And later on, this was sophisticated much more. So this was physiological understanding, but there's also a structural understanding of what's going on. So these are two elements. It's biochemistry, it's important on the one side. And on the other hand, you need the structure and the morphology to get a full picture of what's going on. So in the future, you will always have to have these two aspects next to each other. So, of course, microscopy has helped to understand the function of nerves much better. This was already in the 1600s. There was the first person that made a nice functional microscope. Actually, here in the Netherlands was Anthony van Leeuwenhoek. And this has taken a long way. So these are the first pictures of how the nerve looks when it goes inside of the muscle with the branch structure. This is what I'm going to show to you in a little bit more detail. So this was in 1718, and this is what such a microscope looks like now. We have this one in our lab. So this is very sophisticated and very sensitive, and we can analyze a lot of structures in there, even living neurons. So this is what the neuromuscular junction looks like. These are the nerves, a single axon that extends into the muscle, and here it branches out a lot. And one of the advantages of modern, I think, molecular life sciences is that you can fluorescently label individual proteins and structures. So in this case, there's a genetic construct that labels the nerves with GFP. So this is tremendously important because we can really see what is going on. And it also tells you something functional. I think we need to dim the light a little bit because there's quite some nice immunofluorescent pictures coming up. Okay, I don't. It goes slower than I thought. Oh, the red is really bad. We're going to skip this one because it's much nicer. So you don't see it here in this projector. You can stain some neurotransmitter receptors as well. This is a trick that is difficult to do, but what is possible is to label different nerves with different fluorescent colors. So there's not only GFP, but you also have cyan FP, so there's green and blue fluorescent protein. There's also RFP. And if you use mixtures, you can label actually all the nerves in different colors. Have you ever seen this? It was on this Discovery Channel the other day. And that's why I put it in, because I like it so much. It's not really directly related to any research that we're doing right now, but this is the future of neuroscience to some extent, because you can really visualize all the nerves, in this case in the muscle, all the neuromuscular junctions here, individually and follow them in time as well. This is how the brain is organized when you look at individual neurons stained with CNFP, YFP, that's yellow fluorescent protein, and red fluorescent protein. The trick that they use here is that in each mouse they have 2 alleles, and of these, and they can recombinate at random. So in this way you can make 81 different colors in a mouse brain, and follow in that way. The red has fallen away, unfortunately, in this projector. So you only see 2 colors here. Well, they're actually 3. Come to my computer, I will put the PDF on a layer, you can have a look at this. Now this looks kind of dull compared to the real image. Sorry about that. The effect has gone away. So this is all thanks to the molecular biology of recent years with the Krelok's recombinase system that allows us to put into individual sites of the genome all kind of proteins that we want there. They can be reporter plasmids, like in this case, you just want to know where is what, and they can also really modify the function of a gene. Of course, this system can also be applied to cell lines as well as animals. Yeah, everything that's black here was actually red originally. Maybe it's just to connect. Yes.
Fancy.
That helps, right? Yes, confetti. Yeah. Well, this is what your brain looks like. So all these not as colorful, but they would be colorful if you thought of the activity as well, which is this is one problem that in research you generally don't, or have very few possibilities to follow things over time, which is very important in the future. Typically what people look at is static moments, because you need to sacrifice an animal to get something like this. And the future is that with imaging technologies that are becoming available, you can also follow living animals over time, and even living humans with PET scans, and so there's a huge development there as well. So, but how do you quantify and properly analyze something like this? Of course, this gives you information about structure, and a lot of biologists, at least in the past, were trained to recognize and understand structures. They were counting the petals of a flower and count how many thousand an arthropod has. But in order to properly do science, you need to measure. And how do you measure? How do you quantitate an image like this? What can you do? Well, we have a huge advantage. With light is quantitative. The intensity can be measured. And we do that in lots of assays. For example, ELISAs, Western blot, the fax machine, and we can do it in microscopy as well. You can look at structure, that's this one, that's not so important. And let me go back. Yeah, they are part of research where the quantification is important. So here you've got a neuromuscular junction, I showed it to you before. Here's the nerve, and here there's the presynaptic part of the nerve, and this is the postsynaptic part. In this case, it's a nerve protein that's called vesicular acetylcholine transporter, and this is the acetylcholine receptor. This is the normal situation, and this is when it has gone wrong in an autoimmune disease. So here you can see this protein is still intact, and this protein and another one that's associated, they have gone. So you can imagine if this happens in a nerve, in this case it's in a muscle, there will be loss of function. But how do you quantitate this? First, to understand what's going on in this example, I was showing you a picture of a nerve terminal. Typical nerve terminals have vesicles with neurotransmitter, in this case acetylcholine, and what happens when they get an action potential, they will go near the presynaptic membrane, they will fuse with the membrane in a process called exocytosis, and the acetylcholine will be released. And the protein that I showed you in red before is this acetylcholine receptor that's shown here. And if this happens, these ion channels will open and will permit calcium and sodium to go in and potassium to go out. In one way or another, with other ions maybe, and with other neurotransmitter, most of the neurons in the brain function like this. This is another example where the same thing is going on in the muscle. And what happens then is that other ion channels open after there's a depolarization at this membrane and will induce thereby an action potential. So just in a nutshell how this structure works. How is this caused in a possible in a disease that I'm studying? What happens is that antibodies combine and they can lead to internalization of the receptor in this process, which is an endocytosis. It goes the other way around. And this will lead to the endocytosis and degradation of the receptor. And then there will be fewer here. That's the mechanism of the disease where I showed you before that there were less acetylcholine receptors by immunofluorescence. But again, how do you quantify something like this? This is not what I wanted to show. I'll skip a few slides and I'll do this one. So this is a normal situation. A couple of neuromuscular junctions with the nerve terminal protein in blue, here in green the postsynaptic protein, and here another one, the acetylcholine receptor that I showed you before. And what you can do is you can, using software, freeware software, you're going to use it in this course called ImageJ. Has anybody ever used it or seen it? No. It's a program for image analysis, programmed by the NIH in America. You can download it for free. Try to do so. This name is here. Put it on your computer, and I will show to you how to analyze a dot plot, which is like a Western blot in this course this week. Okay? So I'll send you a link via Liam. Try to download it. If you have problems installing it, we use a computer in my lab to analyze the images. So what you can do, you can outline the edges of such a structure. You can add this region of interest to the program, and then you simply measure the average fluorescent intensity in this area. So we did that for publication, and this works very nicely. You see here an example where we measured that the intensity of, in this case, the acetylcholine receptor per ion plate was normalized to 1 in a control condition, and in a disease, which is here, this has been reduced by 40%, I would say. So that's the average fluorescence intensity. So this is black and white data of something that is qualitative, like going back to, sorry, there were a few extra slides that don't apply here. That's the quantification basically of this. This is an extreme example, but if you average out not only one picture but hundreds, then you will get a quantitative figure. And nothing less, really nothing less you do by using a FACS, fluorescent activated cell sorting. Have you ever heard about that technique? Okay. It's also to analyze fluorescence on a single cell, but basically the same thing as this. And tomorrow we're going to do an ELISA. There we don't use fluorescence as an optical signal, but simply absorption, which is another property of a chemical to not allow light to come through. Right, I had hoped to be able to use a board here for drawing something. Ah, yeah. Let's see when I invented this technique, because it was not known at that time. I submitted a paper with this data, and the review was, well, the use of immunocytokines is not recommended, since fluorescence is a not linear signal that relates directly to the concentration of the epitopes. And of course, that's a nonsense. This is of 5 years ago, and now it's very well accepted. You can use this technique because light and color are very useful in biochemistry. You can quantify light. And where to use it? Here, for example, in a FACS. Here in a Western blot, you can also use fluorescence in a Western blot instead of DAP staining. You can, or ECL. You can also use fluorescent labels. So, and they can be used in immunoassays. This is, I think, a little bit too detailed. It's linear. That's what is shown here. So the light intensity is linear related to this one, to the intensity of the dye on the x-axis. So, can you also quantify structures? Now I was talking about intensity. Yes, also structures can be quantified. Now this is a nerve, this is a muscle fiber, so the nerve is much thinner. I shouldn't say nerve, maybe I should say axon. This is a nerve terminal. When you remove the nerve, you see this. So there's little holes in your muscles where the nerve is embedded into. You can imagine the nerve goes like this into the surface of the muscle fiber. And when you look at that from the side, you might notice these little clefts here, right? They're really called clefts. This is to make the surface much larger. That's where all the neurotransmitters are located. And when you look at it in transmission electron microscopy, you get structures like this. So here you can see the nerve terminal. Here are actually all the vesicles with the neurotransmitter. And here is the postsynaptic membrane. It's all full of clefts, as you see. Postsynaptic folds. It's to make the surface membrane larger. So is this something that could be measured and quantified? The answer is also yes, can be. Just another example. Here I think you can see the vesicles. That's why I put this slide here. We see these round structures. So that's the nerve terminal. That's what you have also in your brains, lots of these. What is specialized in the muscle is that this postsynaptic membrane is completely folded like this. You can see that here are all the acetylcholine receptors in this part, because the membrane is so thick. A normal membrane, just a lipid bilayer in electron microscopy, is very thin, like these ones. If there's proteins located in this bilayer, you get thick membranes like this. Also can be measured, of course. In a disease, what happens? This membrane will be intact, and if autoantibodies come to destruct the acetylcholine receptor, like I showed to you before, then this membrane, the postsynaptic part, will be completely going away. Will be losing all the structure and the folds. This is again a normal situation, all the folds here. This is a nucleus, by the way. Here's a nerve terminal. These are mitochondria. So, nerve terminal here. You can measure the surface of this, of course, also using image shape. You can measure the length of the presynaptic membrane as defined by where it's opposed to the postsynaptic membrane. And you can follow the length of these folds. This is a little bit tedious, but it can be done. So even something like this, that you would at first think this is purely qualitative, if you do a little bit of effort, everything that you can see, you can quantify. And that's what we did. So this is the analysis of hundreds of these structures. And you just need to follow all these membranes and trace them out. And then you can find a significant difference between two conditions that we used in this research. So how are we doing in time-wise? Because I'm still not at the point.
I've got a question. What is rhabsin?
Rhabsin is the rhabsin-associated protein of the synapse. That was the green protein that I showed to you before as the one that's associated with the acetylcholine receptor. I'll show to you a scheme of that. I skipped the slides, and now you ask for it anyway. See what we did here? We try to understand why can the antibodies so easily degrade this receptor? Why is this so easily internalized? And it's not easily internalized if there's this other protein that is anchoring these two together. And this protein is called rhabsin. And what we did there is we overexpressed it in the muscle. And when we added the antibodies, then this protein, the acetylcholine receptor, was stabilized. Since you're asking, I'll use the opportunity to tell you about it anyway. What we did, we injected a plasmid into the muscle where the acetylcholine receptor is localized, and the plasmid is encoding for rhabsin. So this is like transfecting a cell line, but in this case in vivo. And we used electroporation to get the plasmid in. So electric current can be used to make holes in membranes, and it can be used to do plasmapheresis of the plasmid, and then you can get DNA into the cells. And then in this case, we electroporated the the rhabdin inside of the cells, like you see it here, staining green. This is immunofluorescence, so this is not GFP like before, but this is the recombinant protein that is expressed there in a higher concentration than normal. And what it actually did, I'll show you here, no here, is that it's, oh, there was another one. There's a slide missing. What we did, we stabilized all the end plates and all the acetylcholine receptors here. So in the untreated muscle, the acetylcholine receptor was all gone, and in the treated muscles, it was stabilized. So based on.
But it was stabilized. Was it still functional?
Yeah. So we had, unfortunately, we could only do this in one animal. So we had a completely ill animal that was paralyzed because of the antibodies, and one muscle was still working. So it was a gene therapy for one muscle, just to try out if it works. So this is a possible application of DNA technology, is gene therapy. You can use it for understanding, studying structure and function of tissue. And you can also try to use this understanding for designing a therapy, and that's what we did here. But it's not one that can be translated into humans, because you would need to transfect all the muscles. One disease where you really wanted that would be muscular dystrophy, for example.
Okay, but I can imagine like the antibodies, they bind to the receptor, and then you've got rhabdin to stabilize that it doesn't get internalized.
Yeah.
But the receptor is still, there's still something at the receptor, so how can it react to acetylcholine?
The antibodies, you see, if you look at the binding site here, is not the binding site for the acetylcholine. So because these proteins are so huge, some of the antibodies bind to the wrong place and then they block the receptor. But most antibodies bind to a different epitope and they leave the function of the receptor intact. So the effector mechanism in this case would be purely the internalization, which is called antigenic modulation, and the destruction of the postsynaptic folds, but not so much the inhibition of acetylcholine binding or the opening of the channel.
But how does that rhapsody becomes overexpressed? How do you do that?
I do that with.
Is it knock-in or?
It's not really a transgenic technique. It is basically by only injecting a plasmid. Only injecting a plasmid, which is then cytoplasmatically expressed in the tissue. So if you just want to treat one particular cell type, you might as well target only that cell type. So you don't need to go via the germline. You don't need to modify all the cells, but you can just take those cells. For example, there's a group here in the building with Wilfried Germerat that transfects dendritic cells. So you take them out of the body, you modify them, and then you give them back to the patient. So there's techniques to modify, genetically modify cells without putting it in the chromosomes. Actually, that's what happens when you get a virus infection. The virus particles get inside of the cell, they express all their proteins, and you have to understand this in a way that you infected these cells, these muscle cells, with the virus. Basically, what we use are vectors that are based on viral expression vectors. So it's I think it's the SV40 virus. So that's a bit of recombinant DNA. Did you learn about expression systems at all? You know that you can transfect bacteria with plasmids. Then you might know that you can also transfect mammalian cells with plasmids. It's the same way with shuttle vectors. So these bacterial vectors, some of them are modified to put also viruses from humans or mammalian origin, and they will replicate in there to some extent, unless you take these sequences out as well, which we normally do. So instead of using bacteria, you can also use bacterial vectors. You can use also eukaryotic vectors, and they will be functional in our tissues. So one example that is probably used at the moment is you know these cyclists that they take EPO to get up the mountain a little bit faster, right? To have more hemoglobin and so on. and they can't take recombinant EPO anymore because that's detected by all the assays. So what they're doing is they use the gene encoding for EPO and put it in a plasmid and electroplate it in their muscle, and that works. So then they produce their own EPO, not in the normal way, but in their muscles directly. It's illegal, of course. But I've heard that is also detectable because the glycosylation patterns are the same as recombinant EPO, so it's not very useful. But I've read something that this has been tried out.